Suppose now that other channels permeable to some other ion, say Na+, are suddenly opened in the plasma membrane. Because Na+ is at a higher concentration outside the cell than inside, Na+ will move into the cell through these channels, and the membrane potential will become less negative, maybe even reversing sign to become positive, so that the interior of the cell is positive with respect to the exterior. The membrane potential will shift toward a new value that is a compromise between a negative value that would correspond to equilibrium for K+, and the positive value that would correspond to equilibrium for Na+. Any change in the membrane's permeability to specific ions, that is, any change in the number of ion channels of different sorts that are open, thus causes a change in the membrane potential. The membrane potential, therefore, is determined by both the state of the ion channels in the membrane and the ion concentrations in the cytosol and extracellular medium. Because the electrical processes at the plasma membrane occur very quickly compared with changes in the bulk ion concentrations over the short term, milliseconds as compared with seconds or minutes, it is the ion channels that are most important in controlling the membrane potential. To see how the interplay between the membrane potential and ion channels is used for electrical signaling, we now turn from the behavior of ions and ion channels to the behavior of entire cells. We take nerve cells as our prime example, for they, more than any other cell type, have made a profession of electrical signaling and employ ion channels in the most sophisticated ways. Ion channels and signaling in nerve cells. The fundamental task of a nerve cell or neuron is to receive, conduct, and transmit signals. Neurons carry signals inward from sense organs to the central nervous system, the brain and spinal cord. In the central nervous system, neurons signal from one to another through networks of enormous complexity, allowing the brain and spinal cord to analyze, interpret, and respond to the signals coming in from the sense organs. From the central nervous system, neurons extend processes outward to convey signals for action to muscles and glands. To perform these functions, neurons are often extremely elongated. The motor neurons in a human that carry signals from the spinal cord to a muscle in the foot, for example, may be a meter long. Every neuron consists of a cell body containing the nucleus that has a number of long, thin extensions radiating outward from it. Usually, a neuron has one long axon, which conducts signals away from the cell body toward distant target cells. It also usually has several shorter, branching dendrites, which extend from the cell body like antennae and provide an enlarged surface area to receive signals from the axons of other neurons. Figure 1231. The axon commonly divides at its far end into many branches, each of which ends in a nerve terminal, so that the neuron's message can be passed simultaneously to many target cells, either other neurons or muscle or gland cells. Likewise, the branching of the dendrites can be extensive, in some cases sufficient to receive as many as 100,000 inputs on a single neuron. No matter what the meaning of the signal a neuron carries, whether it is visual information from the eye, a motor command to a muscle, or one step in a complex network of neural processing in the brain, the form of the signal is always the same. It consists of changes in the electrical potential across the neuron's plasma membrane. Action potentials provide for rapid long-distance communication. A neuron is stimulated by a signal, typically from another neuron, delivered to a localized site on its surface. This signal initiates a change in the membrane potential at that site. To transmit the signal onward, however, the change in membrane potential has to spread from this point, which is usually on a dendrite or the cell body, to the axon terminals, which relay the signal to the next cells in the pathway. Although a local change in membrane potential will spread passively along an axon or a dendrite to adjacent regions of the plasma membrane, it rapidly becomes weaker with increasing distance from the source. Over short distances, this weakening is unimportant, but for long-distance communication, such passive spread is inadequate. In the same way, a telephone signal can be transmitted without amplification the short distances through the wires in your hometown, but for transmission across an ocean by an undersea cable, the strength of the signal has to be boosted at intervals. Neurons solve this long-distance communication problem by employing an active signaling mechanism. A local electrical stimulus of sufficient strength triggers an explosion of electrical activity in the plasma membrane that is propagated rapidly along the membrane of the axon and sustained by automatic renewal all along the way. This traveling wave of electrical excitation, known as an action potential or a nerve impulse, can carry a message, without the signal weakening, from one end of a neuron to the other at speeds of up to 100 meters per second. All of the early research that established the mechanism of electrical signaling along nerve axons was done on the giant axon of the squid. Figure 1232. This axon has such a large diameter that it is possible to record its electrical activity from an electrode inserted directly into it. How we know, pages 412 to 413. From such studies, it was deduced that action potentials are the direct consequence of the properties of voltage-gated ion channels. See figure 1225a in the nerve cell membrane, as we now explain. Action potentials are usually mediated by voltage-gated Na+ channels. An action potential in a neuron is typically triggered by a sudden local depolarization of the plasma membrane, that is, by a shift in the membrane potential to a less negative value, that is, a shift towards zero. We discuss later how such a depolarization is caused by the action of signal molecules, neurotransmitters, released by another neuron. A stimulus that causes a sufficiently large depolarization to pass a certain threshold value promptly causes voltage-gated Na+ channels to open temporarily at that site, allowing a small amount of Na+ to enter the cell down its electrochemical gradient. The influx of positive charge depolarizes the membrane further, that is, it makes the membrane potential even less negative, thereby opening more voltage-gated Na+ channels, which emit more Na+ ions and cause still further depolarization. This process continues in a self-amplifying fashion until, within about a millisecond, the membrane potential in the local region of membrane has shifted from its resting value of about -60 millivolts to about +40 millivolts. Figure 1233. This voltage is close to the membrane potential at which the electrochemical driving force for movement of Na+ across the membrane is zero, that is, at which the effects of the membrane potential and the concentration gradient for Na+ are equal and opposite, and Na+ has no further tendency to enter or leave the cell. If the channels continue to respond indefinitely in this way to the altered membrane potential, the cell would get stuck at this point with all of its voltage-gated Na+ channels predominantly open. The cell is saved from this fate because the Na+ channels have an automatic inactivating mechanism, which causes them to rapidly adopt, within a millisecond or so, a special inactive conformation, where the channel is unable to open again. Even though the membrane is still depolarized, the Na+ channels will remain in this inactivated state until a few milliseconds after the membrane potential returns to its initial negative value. A schematic illustration of these three distinct states of the voltage-gated Na+ channel closed, open, and inactivated, is shown in Figure 1234. How they contribute to the rise and fall of the action potential is shown in Figure 1235. The membrane is further helped to return to its resting value by the opening of voltage-gated K+ channels. These also open in response to depolarization of the membrane, but not as promptly as the Na+ channels, and they then stay open as long as the membrane remains depolarized. As the action potential reaches its peak, K+ ions carrying positive charge therefore start to flow out of the cell through these K+ channels down their electrochemical gradient, temporarily unhindered by the negative membrane potential that restrains them in the resting cell. The rapid outflow of K+ through the voltage-gated K+ channels brings the membrane back to its resting state more quickly than could be achieved by K+ outflow through the K+ leak channels alone. The description of an action potential concerns only a small patch of a plasma membrane. The self-amplifying depolarization of the patch, however, is sufficient to depolarize neighboring regions of membrane, which then go through the same self-amplifying cycle. In this way, the action potential spreads outward as a traveling wave from the initial site of depolarization, eventually reaching the extremities of the axon. Figure 1239. Faced with the consequences of the Na+ and K+ fluxes caused by a passing action potential, local Na+/K+ ATPase molecules labor continuously to restore the ion gradients across the plasma membrane of the axon. Voltage-gated Ca2+ channels convert electrical signals into chemical signals at nerve terminals. When an action potential reaches the nerve terminals at the end of an axon, the signal must somehow be relayed to the target cells that the nerve terminals contact, usually neurons or muscle cells. The signal is transmitted to the target cells at specialized junctions known as synapses. At most synapses, the plasma membranes of the transmitting and receiving cells, the presynaptic and the postsynaptic cells respectively, are separated from each other by a narrow synaptic cleft, typically 20 nanometers across, which the electrical signal cannot cross. Figure 1240. For the message to be transmitted from one neuron to another, the electrical signal is converted into a chemical signal in the form of a small signal molecule known as a neurotransmitter. Neurotransmitters are stored ready-made in the nerve terminals, packaged in a membrane-enclosed synaptic vesicles. See Figure 1240. When the action potential reaches the terminal, the neurotransmitters are released from the nerve endings by exocytosis, discussed in Chapter 15. This link between the action potential and secretion involves the activation of yet another type of voltage-gated cation channel. The depolarization of the nerve terminal plasma membrane caused by the arrival of the action potential transiently opens voltage-gated Ca2+ channels, which are concentrated in the plasma membrane of the presynaptic nerve terminal. Because the Ca2+ concentration outside the cell is more than 1,000 times greater than the free Ca2+ concentration in the cytosol, Ca2+ rushes into the nerve terminal through the open channels. The resulting increase in Ca2+ concentration in the cytosol of the presynaptic cell triggers the fusion of some of the synaptic vesicles with the plasma membrane, releasing the neurotransmitter into the synaptic cleft. Thanks to the voltage-gated Ca2+ channels, the electrical signal has now been converted into a chemical signal. Figure 1241. Transmitter-gated channels in target cells convert chemical signals back into electrical signals. The released neurotransmitter rapidly diffuses across the synaptic cleft and binds to neurotransmitter receptors concentrated in the postsynaptic membrane of the target cell. The binding of neurotransmitter to its receptors causes a change in the membrane potential of the target cell, which can trigger the cell to fire an action potential. The neurotransmitter is then quickly removed from the synaptic cleft, either by enzymes that destroy it, or by reuptake into the nerve terminals that released it, or into neighboring cells. This rapid removal of the neurotransmitter limits the signal and ensures that, when the presynaptic cells fall quiet, the postsynaptic cell will fall quiet as well. Neurotransmitter receptors can be of various types. Some mediate relatively slow effects in the target cell, whereas others trigger more rapid responses. Rapid responses on a timescale of milliseconds depends on receptors that are transmitter-gated ion channels, also called ion channel coupled receptors. These constitute a subclass of ligand-gated ion channels. See Figure 1225b, and their function is to convert the chemical signal carried by neurotransmitter back into an electrical signal. The channels open transiently in response to the binding of the neurotransmitter, thus changing the ion permeability of the postsynaptic membrane. This in turn causes a change in the membrane potential. Figure 1242. If the change is big enough, it can trigger an action potential in the postsynaptic cell. A well-studied example of a transmitter-gated ion channel is found at the neuromuscular junction, the specialized type of synapse formed between a neuron and a muscle cell. In vertebrates, the neurotransmitter here is acetylcholine, and the transmitter-gated ion channel is the acetylcholine receptor. Figure 1243. Neurons receive both excitatory and inhibitory inputs. The response produced by a neurotransmitter at a synapse can be either excitatory or inhibitory. Excitatory neurotransmitters delivered by axon terminals or of excitatory neurons stimulate the postsynaptic cell, encouraging it to fire an action potential. Inhibitory neurotransmitters delivered by axon terminals of inhibitory neurons do the opposite, discouraging the postsynaptic cell from firing. The drug curare, which South American Indians used to make poison arrows and surgeons use to relax muscles during an operation, causes paralysis by blocking the delivery of excitatory signals at neuromuscular junctions. By contrast, strychnine, a common ingredient in rat poisons, causes muscle spasms, convulsions, and death by blocking the delivery of inhibitory signals. Excitatory and inhibitory neurotransmitters bind to different receptors, and it is a character of the receptor that makes the difference between excitation and inhibition. The chief receptors for excitatory neurotransmitters, mainly acetylcholine and glutamate, are ligand-gated cation channels. When a neurotransmitter binds, the channels open to allow an influx of cations, which depolarizes the plasma membrane toward the threshold potential required for triggering an action potential. Stimulation of these receptors thus tends to activate the postsynaptic cell. The receptors for inhibitory neurotransmitters, mainly aminobutyric acid, GABA, and glycine, by contrast, are ligand-gated Cl- channels. When a neurotransmitter binds, the channels open, but very little Cl- enters the cell at this point because the driving force for movement of Cl- across the membrane is close to zero at the resting membrane potential. If an excitatory neurotransmitter opens Na+ channels at the same time, however, the resulting depolarization caused by the Na+ influx will cause Cl- to move into the cell through the open Cl- channels, neutralizing the effect of the Na+ influx. Figure 1244. In this way, inhibitory neurotransmitters suppress the production of an action potential by making the target cell membrane much harder to depolarize. The locations and functions of these ion channels and of some of the other ion channels discussed in this chapter are summarized in Table 12.3. Transmitter-gated ion channels are major targets for psychoactive drugs. Most drugs used in the treatment of insomnia, anxiety, depression, and schizophrenia exert their effects at synapses in the brain, and many of them act by binding to transmitter-gated ion channels. The barbiturates and tranquilizers such as Valium, Ambien, and temazepam, for example, bind to GABA-gated Cl- channels. Their binding makes the channels easier to open by GABA, thus making the cell more sensitive to GABA's inhibitory action. By contrast, the antidepressant Prozac blocks the reuptake of an excitatory neurotransmitter, serotonin, increasing the amount of serotonin available at those synapses that use this transmitter. Why this should relieve depression is still a mystery. The number of distinct types of neurotransmitter receptors is very large, although they fall into a small number of families. There are, for example, many subtypes of acetylcholine, glutamate, GABA, glycine, and serotonin receptors. They are usually located in different neurons and often differ only subtly in their properties. With such a large variety of receptors, it may be possible to design a new generation of psychoactive drugs that will act more selectively on specific sets of neurons to alleviate the mental illnesses that devastate so many people's lives. 1% of the human population, for example, have schizophrenia. Another 1% have bipolar disorder, and many more suffer from anxiety or depression. Synaptic connections enable you to think, act, and remember. At a synapse, the nerve terminal of the presynaptic cell converts an electrical signal into a chemical one, and the postsynaptic cell converts the chemical signal back into an electrical one. Interference with these processes, for good or ill, is of enormous practical importance to us. But why has evolution favored such an apparently inefficient way to pass on an electrical signal? It would seem more efficient to have a direct electrical connection between the pre- and postsynaptic cells, or to do away with the synapse altogether and use a single continuous cell.